Hi there, in this lecture we will be discussing the different types of transport occurring across the cellular membranes. So, different kinds of transport which occur across the cellular membrane include diffusion. This diffusion can be either simple diffusion or it can be facilitated diffusion. Other is active transport, and the third one endocytosis. Fourth one is exocytosis. So we will discuss them one by one. So diffusion is along concentration gradient. It is a movement along concentration gradient and no energy consumed here. No energy consumed. Okay. While active transport is against concentration gradient, and energy is consumed here. Okay, so let's first discuss diffusion. So what is diffusion here? It is positively a cellular membrane. So diffusion is the simple process in which lipid soluble substances cross the cellular membrane from their higher concentration to lower concentrations. This is simple diffusion in which substances which are for which the membrane is permeable cross through the membrane. This is simple diffusion. However, for the positive ions, for example K positive, potassium or sodium positive, there is a requirement of some carrier protein. So they cross the membrane with the help of some carrier protein. Okay, this is a carrier protein. Sodium will bind here. This is a pink state, and then it will undergo conformational change, and then it will transfer sodium to the inner aspect, which is Pong state. Okay, so this carrier protein will fluctuate between pink state, in which it will bind to some external molecule, and then it will undergo conformational change to Pong state and will transfer that molecule to the inner aspect of cellular membrane. So this is facilitated diffusion. Next is active transport. What is active transport? In active transport, molecule moves from lower concentration positively. This is membrane here. Okay, and there is a substance. Supposedly sodium is here, and it is here in its lower concentration. Let's take example of potassium. For example, it's in lower concentration here, and here it is higher concentration of potassium. So this potassium will move from its lower to higher concentration with the help of some protein, and this protein will consume the energy from ATP, and this transport will be active transport as it is consuming energy. Or it can be in the form of sympoters. What are sympoters? Let me explain the sympoters. Sympoters are the antipoters. For example, in certain membranes, molecule may move in the opposite direction or in the same direction with the help of one carrier protein. For example, here is a carrier protein. Here, and it has two binding sites. On one side binds glucose, and on other side binds sodium. Sodium moves along its concentration gradient. Sodium moves from higher concentration to lower concentration, and in this movement it releases energy. And this glucose is moving from low concentration to higher concentration, and in this uphill movement energy is required. So this energy comes from the downhill movement of sodium. So sodium, this carrier protein has two binding sites, one for sodium, one for glucose. Sodium moves along its concentration gradient and releases energy, while glucose moves against concentration gradient and consumes energy. So this is the example of symport. And what happen in antipode transport? In antipode transport, substances move in the similar way but in opposite direction. For example, if glucose binds here and sodium moves along concentration gradient from this side to this side, while glucose moves from its low concentration to higher concentration from here, then it is an example of antipode, in which again Downhill movement of sodium releases energy which is used by uphill movement of glucose, but as they both are moving in opposite direction, so this is an example of antipode transport. In this transport there is also consumption of energy. So these are two kinds of transport which occur across the cellular membrane. Next is endocytosis and exocytosis. What is endocytosis? This is, for example, any substance here, the cell comes and cell want to eat it puts its pseudopods around it and forms a vesicle, and then this vesicle is processed either by Golgi apparatus and endoplasmic reticulum, or it fuses with lysosomes and is digested. So this is an example of endocytosis. So what is exocytosis? In exocytosis, cell releases some molecule. For example, this is a cell here and here is some vesicle. It comes here, it fuses with the cellular membrane and pours its substances outside, as we study in the release of acetylcholine at cellular at synapses and so on. So it is an example of exocytosis. Okay. So these are this endocytosis. This is the general concept of endocytosis and exocytosis. And this endocytosis, it can be phagocytosis. Phagocytosis if there is eating of solid materials, and it can be pinocytosis if there is eating of the liquid materials. Okay. And this pinocytosis can be general, in which there is no proper receptors involved, or it can be receptor specific. In general, it generally puts pseudopods around the material and eats it up, while in receptor mediated cell have certain receptors, and the substance which is going to be pinocytosed comes here in interaction with the receptor, and the receptor is internally coated by a protein called clathrin. This protein pinches and forms clathrin coated vesicle. It forms clathrin coated vesicle, which happens in the receptor mediated or specific pinocytosis. So it is an overview of different kind of transports which occur across the cellular membranes. Another concept which we need to know here is whenever there is diarrhea, there is dehydration. So to counter this dehydration, oral rehydration therapy is done, in which ORS are given, and they are usually given in the form of glucose plus NaCl. So what happens? Glucose and NaCl both goes to the intestine and intestinal cell. There are sympotors. Of, this is lumen of intestine and this is intestinal cell. There are sympotors. Sodium binds here and glucose binds here, and sodium moves along concentration gradient while glucose move against concentration gradient, and both enter the intestinal cell, and then from there they diffuse to the blood. And here there is a specific glucose transporter is GLUT2. So they diffuse to the blood, and when they diffuse to the blood, water follows them, as solutes are going here, so water follows the solute. So there is reabsorption of water, and there is cure of diarrhea, plus dehydration is also countered. So rehydration occurs, phenomena of rehydration occurs. So it is how sodium and glucose are given for rehydration in people with diarrhea. What happens? Glucose and sodium both bound to their symphotor, then they move to the intestinal cell. From intestinal cell they diffuse to the blood, and water also follows them, and the process of rehydration occurs. If sodium is given alone or the glucose is given alone, then the benefit will be much less as compared to their combined preparation. Okay. Another thing we need to know here is the lipid raft and caveole. So what are these? Lipid raft is basically an area of this is, for example, cellular membrane, an area of cellular membrane where different glycoproteins and there are different glycoproteins as well as integral proteins and cholesterol and sphingolipids are concentrated, and it is a concentrated area with different kinds of lipids and proteins, and it plays its significant role into the signal transduction. This specific area is called lipid raft and plays its role in signal transduction, and some bacteria may misuse it by binding to receptor and can gain entry into the cell by lipid rafts. So what is caveole? Caveole is an indentation into the cellular membrane, and this indentation also contains similarly different proteins and receptors. Okay, and it also played its role. It also plays its role in signal transduction, and some bacteria can also misuse it to gain entry into the cell. So these are lipid raft and caveoles. So we have discussed different transport mechanism across the cellular membranes. Two more things we should discuss here are the aquaporins and ionophores. So what are aquaporins and what are ionophores? Aquaporins are basically specific proteins. Aquaporins are basically specific proteins which form water channels. They form water channels and allow the water to diffuse across the cellular membrane. They form water channels and allow the diffusion of water. However, the positive hydrogen ions cannot, large ion sodium, potassium cannot cross these aquaporins. Moreover, hydrogen cannot cross these aquaporins because water is crossing here, and to diffuse along with the water, these ions need to interact with the partially negative charge oxygen. However, the aquaporin proteins have aspartate residues here, and this partial negative charge oxygen of water is interacting with these aspartate residues. So it does not find any space or any opportunity to interact with hydrogen or other positive ions. So it does not allow to cross the ions. So aquaporin channel allow water molecule to cross it. However, if there is some mutation in aquaporin protein, especially aquaporin protein 2, it can lead to the nephrogenic diabetes insipidus, in which water channels are not sensitive to the hormones. As a result, there is inability to concentrate the urine. Next thing we need to know is ionophores. So what are ionophores? Ionophores, as the name shows, the ionophores are specifically pores in the cellular membrane that allows ions and metabolites to cross the cellular membrane. So what happens is, positive, this is a cell here, and different kinds of toxins as well as drugs and certain bacteria can create specific pores in the cellular membrane and can form specific channels which are called ionophores. And what these channels do, they can cause the movement of potassium outside the cell, or they can cause movement of ATP outside the cell or other cellular material to diffuse outside the cell, and what it causes can lead to the cellular death due to depletion of these essential cellular materials. So these are ionophores. A typical example of this ionophore is alpha hemolysin toxin of streptococcus. This toxin creates a channel here in the cellular membrane, which leads to the diffusion of ATP outside of the cell and lead to the cellular lysis, of cells. So it was a concept of aquaporins and ionophores. Okay, so we have discussed different transporters and different kind of channel protein and so on. These channels are very important. Their mutation can lead to the different diseases. For example, a common disease is cystic fibrosis, which is due to defect in channel cystic fibrosis transmembrane regulator. This channel is basically a transporter of choline. So this defect leads to the increased choline in sweat, as well as it can lead to the infertility in males. It can lead to the fat maldigestion due to pancreatic insufficiency. It can lead to the airway infections and so on. So, and any defect in the working of a brain channel can lead to the multiple issues, and there are multiple diseases which arise due to defect in these channels. So the reference material for this lecture is also again from the chapter 40 of Harper's Biochemistry. Thank you.